try to get the recording started. Okay, I think we'll get started. Thanks everybody for coming. Really happy to introduce our guest speaker for the Marine Science Seminar, is Eric Galbraith. And he is a full professor at McGill in the Department of Earth and Planetary Science. He's also an alumni of that department where he did his bachelor's. I believe he's originally from Halifax. And he did his PhD at the University of British Columbia. And I first met Eric actually when I was a graduate student at the Lamont Doherty Earth Observatory, and Eric was doing a postdoc at Princeton GFDL, and he had come to give a talk. So, these seminars are a good way to meet new colleagues. So Eric has also worked at the Autonomous University in Barcelona for several years, and he's been successful in attaining a lot of research funding in 3 countries. So that's good. And he has a lot of research and publications that are highly cited in paleoceanography, the global nitrogen cycle, and modern climate assessments. And more recently, he's been focusing on some of the more uniquely human problems in the Earth system, like fisheries, carbon pricing, and global happiness. So we're all happy that he's here with us today and going to be talking about fisheries and the iron cycle. So let's welcome Eric and take it away.
Great. Thanks very much, Chris. And thanks for the invitation. It's nice to virtually be here. So, right, as Chris alluded to there, I've been working on a number of topics recently. Seems to be an exponential growth in topics that I'm working on. And I have to admit, I haven't been working on this one in particular in a little while. So I'm a little tiny bit rusty on it, but I know Chris is a big fan of the iron cycle, and I thought it would be fun to talk about this with your group. So, I'll try to go through it fairly quickly because I know Zoom is difficult to focus on for long periods of time. But please, if you would like to interrupt me at any time, please try to do so. I'm always happy to take questions in the middle if they come up. Okay. So, right, so I'm really going to talk about work that's published in 2 papers that I did, mostly with Priscilla Lemezzo, who's shown here. She's currently in Paris, but also David Kreutzma and Daniele Bianchi were very important collaborators for this work as well. Oops, what's happened now? Okay, so in the world of fish ecology, it's widely recognized that there are strong environmental controls on fish abundance. These are usually taken to be primary production. Sorry, can you see that Bruce Mackenzie entered the waiting room there? Is that in the middle of the screen? No, that's hidden. Okay.
I'll take care of that. Yeah.
Okay. So it's usually widely recognized that primary production can limit fish growth. Also, water temperature plays a role. Things growing faster in warmer water. Benthic habitat type is obviously important, and the water depth related to that. Oxygen concentrations are seen as a big control on fish abundance, particularly now that oxygen concentrations are changing rapidly. And even pH is often included as an important control on fish, especially larval fish growth. In a world away, organisms kind of conversely are thought to feed back on the environment, including iron cycling. But the organisms that are usually thought of as influencing iron cycling are generally limited to phytoplankton and zooplankton. And then, of course, bacteria, if we kind of think of as being all over the place. So what I'm going to try to convince you of today is that the iron cycle is missing as an important control on wild fish distributions in the ocean, and that fish should also be included in the iron cycling. kind of paradigm of how we think about how iron cycles in the ocean. So we'll see if I can succeed in convincing you of those two things. So first, I'm going to talk very, very briefly about iron in the ocean and how it is known to limit plankton growth. Then I'll talk about these relatively limited amount of information we have on fish and iron. I'm then going to make kind of an important detour into the global distribution of fishing effort, what this can tell us about the relationship between iron and fish. And then I'll talk about the influence of fish that we would expect to see on the sinking flux of iron based on some theoretical considerations, and then a little bit of speculation at the end. So first, this is one way to look at the relative abundance of nutrient elements in the ocean. So this plot is showing the average availability, the average dissolved seawater concentration of all of these different elements shown by their elemental abbreviations here in the vertical, and in the horizontal, the cellular abundance of these different elements normalized to carbon. So basically, as you go up vertically on this plot, you get elements that are more abundant in the ocean. And as you go across, you get elements that are more abundant in phytoplankton. So this kind of defines a diagonal above which things are not limiting because they're available in excess on average in the ocean. And below that diagonal, things become more limiting. And I just want to point out here that iron is really in the most limiting position. So relative to how much it's needed, it's quite scarce in the ocean in general. Of course, this is looking at a whole ocean average. The ocean's always circulating, moving nutrients around, and so they're not equally abundant in all different parts of the ocean. So if we look at the distribution of nitrate, so the most abundant form of nitrogen at the surface ocean where there's sunlight and so phytoplankton can grow, we see it has these very large patterns where it's quite abundant in some regions and low abundance in other regions. And these large nitrate-rich regions have become known as high nitrate, low chlorophyll regions in the surface ocean. I don't want to go into detail about those, but just to say, it's been very well shown that if you go out in a ship and you take some water from these regions, or conversely do in situ experiments where you add nutrients, when you go to the nitrate limited regions, you need to add nitrogen in order to see accelerated phytoplankton growth. But when you go to the nitrate rich regions, iron is what needs to be added in order to accelerate the growth of phytoplankton. So these HNLC, nitrate rich regions in the surface ocean, are clearly iron limited. They're not the only iron limited regions in the surface, but these HNLC regions are clearly iron limited. And that's important, so I'll come back to that. The HNLC regions. So phytoplankton can clearly be limited. They can clearly, grow at less than the maximum overall community phytoplankton growth rate if iron is scarce. But at the same time, they're actually quite flexible in their iron stoichiometry, in the iron to carbon ratio that they can have within their cells. And so that's illustrated on this plot. So this plot is showing the iron to carbon concentration of phytoplankton in units that I'll keep using. It's very confusing that iron to carbon ratio is different fields. Everybody uses their own units. But so here I'll use throughout this ratio, which is micromoles iron to mole carbon. So these numbers will show up in other plots as well. But so basically, in these units, the iron content of phytoplankton ranges between something like 2 up to hundreds. So 2 orders of magnitude variation in how iron-rich phytoplankton can be based on essentially the taxonomic group. So this is showing different taxonomic groups. Each taxonomic group has its own particular range of iron richnesses. And then those go along with different environments. So you find different taxonomic groups in different environments. And then within the taxonomic groups, you have environmental dependencies. So quite a bit of flexibility in the iron richness of phytoplankton. Here's just another way of looking at that same thing. So I'll show more plots that look like this later, but so this is showing the iron richness on the horizontal now, and just comparing iron rich waters, which have high iron to carbon ratios in phytoplankton, compared to iron limited waters, which have low iron to carbon in phytoplankton. And so then we can think of the, so this is now just another view of the global surface of the ocean. Now this is showing ocean color. So basically it's green where there's lots of phytoplankton growing. What you see here is that the HNLC regions, which include the subarctic Pacific and the Southern Ocean, the Eastern tropical Pacific, they actually have quite a bit of phytoplankton. You know, they're fairly green. So these are iron limited regions, but it's not fair to think of them as not having phytoplankton. They actually have quite a bit of phytoplankton. It's just that it's iron poor. So these regions have iron poor phytoplankton. So relatively abundant, but iron poor phytoplankton. Okay, so what about the rest of the ecosystem? So if we move up the trophic web from phytoplankton to zooplankton, we can just kind of think generally about what should happen as consumers eat some food source that has some iron to carbon ratio. As they process it and turn it into their own tissues and lose some things through excretion and ingestion, there's going to be a transformation of that stoichiometry. So I don't know if this is an attempt to be a simple diagram, then it got a little complicated. But this is showing a food source with orange iron and blue carbon, which is being ingested by some Pac-Man generic consumer. And then the food is passing through a gut. So really thinking about animals with guts here mostly. And then across that gut wall, there's absorption of the two elements with differing amounts that depend on complicated gut chemistry and gut wall characteristics. And then the rest of it that's not absorbed, it goes into the ingestion pool. And then some of what is absorbed is excreted, and the rest is assimilated into the body tissues. Well, that makes sense. So maybe, well, so you can define a trophic assimilation efficiency, tau x, which would be what's ingested minus what's excreted and egested, divided by what's ingested. So for carbon, this is usually called the trophic efficiency. But you can define the same thing for iron if you want to. So it's basically just how much ends up going into the tissues of the consumer compared to what it ate. And then you can also define an assimilation, which would be the absorption over the ingestion. So if we think about this for zooplankton, that means that the iron to carbon of the zooplankton is going to equal the iron to carbon of the phytoplankton food source times a ratio, which is the overall assimilation for iron compared to the overall assimilation for carbon. Now, if you look at the overall assimilation of carbon, this tends to be something like 0.15 to 0.3 for zooplankton. So it's basically of the food that they eat, that much of it actually contributes to their own tissue growth, and the rest of it is lost for metabolism, to support metabolism. The carbon's burned up. So how much of the iron is absorbed? Well, it turns out that when people looked at this decades ago and found that when you look at the iron assimilation in zooplankton, it tended to follow the iron concentration in the cytoplasm of the plankton that they were eating. So right away, people saw, and this was followed up on a bit with later measurements, but people saw that it's basically not strongly homeostatically controlled. So zooplankton just seemed to follow along with the phytoplankton iron to carbon ratio. This is not true for nitrogen to phosphorus ratios in zooplankton. They're much more fixed, much more homeostatically controlled. But it seems like the zooplankton just go with the flow, basically. If the iron content of the phytoplankton is low, they get a low iron content as well. And you can see that in field measurements. So here this is showing phytoplankton and zooplankton and iron-rich waters. The distributions of the iron contents overlap with each other. And then when you go to iron-limited waters, you find that the zooplankton have low values corresponding with the phytoplankton. Right? So basically, the zooplankton iron to carbon follows the phytoplankton iron to carbon. Can zooplankton be iron limited? We wondered about this and we looked, and of course there was a paper. Can copepods be limited by the iron content of their food from 2011? And it seems the jury is still out. It's hard to identify strong iron limitation of zooplankton from what I've seen when I went through this a couple of years ago. So it seems possible, but there's no signs of very strong iron limitation of zooplankton communities. And regardless, they're abundant in HNLC regions. So now here I'm drawing the HNLC regions with these blue lines, and the colored dots here are showing mesozooplankton biomass from the copepod database. So these are where nets have been trawled through the ocean, the zooplankton has been added up. And you can see that in the HNLC regions, zooplankton are very abundant. Actually, some of the highest mesozooplankton abundances in the world occur in these HNLC regions. So there's definitely lots of iron poor zooplankton in the HNLC regions, quite abundant. Okay, right, but I'm here to talk about fish. So what about the fish? So when we think of the iron to carbon of the fish, if they're eating zooplankton, then we would expect the iron to carbon ratio of the zooplankton to be multiplied by the assimilation ratio for fish in order to give you the iron to carbon ratio of the fish. The assimilation of carbon from fish is not very different, something like 0.15 to 0.2, ranges a bit more than that, but those would be kind of typical values that you would get. What about the assimilation? Are they good or bad at assimilating iron? Well, it turns out, I didn't know this before, but so fish have quite alkaline guts. Teleosts. Teleosts have alkaline guts as part of their salt, their osmotic regulation strategy. They dump lots of bicarbonate into their guts. And if you know something about iron chemistry, that tends to give you complexes of iron, which are then difficult to absorb across the gut wall. So it would appear that fish may be relatively poor at assimilating iron because of their gut chemistry. And the available measurements that we could find tended to be below 0.14. So where people had actually measured the absorption of iron, which is just really a small handful of studies, but they showed quite low values for the absorption efficiency of iron in fish. So when you put together that absorption efficiency with the carbon assimilation efficiency, it suggests a value of less than 1. So that means that the tissues constructed by the fish should be something equal to or lower than the iron to carbon ratio of the zooplankton, right? So this would imply that the food source of the fish needs to be at least as rich in iron as their own tissue requirements. But of course, maybe it's just the fish don't really need much iron. Could be the fish don't eat a whole lot of iron. So we went and looked into that, and not much data. There really was not a lot of data on whole fish iron to carbon ratios. Just a few studies, mostly on salmon, and a couple from a Japanese study on Southern Ocean fish from the 80s. I don't know, they could do it, so they measured it. But what they showed was basically a range from about 10 to 40 micromoles iron per mole carbon. So in these same units. We found a lot more data on nutritional contents of fish, for diet, like human diet studies. Of course, this is just the edible parts of fish, but then we found some studies that compared the edible parts of fish with the whole fish. And so we tried to estimate what the whole fish values would be from the nutritional data. So essentially the muscle of the fish. And those gave a range like this. So where the light colors are showing with a very low whole fish to muscle multiplier ratio, and the dark colored circles here are assuming high. So that's just kind of bracketing the uncertainty in this transformation from muscle to whole fish. But basically the more likely bar would be shown there in blue. So again, going from something like 10 up to as high as potentially 100 in some of the more iron-rich fish. So you can see looking at this figure, that it looks like the marine fish are not lower in iron than zooplankton in general, and definitely not in iron-rich waters. There's no question, I think, that a lot of fish, maybe some fall into the same range, but definitely a lot of fish have significantly higher iron requirements than the available food, going through this line of reasoning. So if this is true, if fish can be limited by iron, you might expect that this would have shown up before in something like aquaculture, where people are trying to grow fish very carefully in cultures. And in fact, it was pointed out in the 1970s that iron can limit fish growth in aquaculture. And therefore, there have been recommendations of how much iron to put in your fish feed for many decades. And it's been shown that the hemoglobin concentrations in the fish drop if they don't have sufficient iron, and the slowest growth can lead to problems in your aquaculture tank. And so we found all the recommended aquaculture feed iron content recommendations that we could, and put them in the same units on this plot. And they all come up at what's shown here in this red bar, so relatively iron rich and similar to the zooplankton concentrations in iron rich water, but up to an order of magnitude higher than the iron concentrations of zooplankton in iron limited waters. So again, consistent with iron not being sufficient in the plankton of iron limited waters. So then finally you could say, okay, maybe it slows their growth a little bit, but does it really affect their abundance? Maybe they're still abundant anyway. And this is a hard question to answer, but so this is where, because really, well, I'm going to keep focusing on these HNLC regions, because not because they're the only iron limited regions, but because they're fairly unambiguously iron poor, and they're easy to mask out. It's easy to say on a global map where they are. So we want to compare HNLC regions as being iron limited versus non-HNLC regions. And we would like to say something about the whole fish community, because it could be that some fish don't have high iron requirements and they can just substitute for the other ones when iron is scarce. But direct measurements of fish abundance are even more scarce. It's very hard to sample them. And the best way to get a good sense of all the fish that are out there is actually to use fishing effort. So this has been done a number of times before. People have used records of fishing and fish catch in order to infer how much fish there are in the water. And thanks to Leonardo DiCaprio, who contributed some funding, Leo to the rescue, there is something called Global Fishing Watch. I don't know if you're all aware of this, but this started a few years ago. So this is actually in collaboration with Google. They listen to the automatic transmissions from all kinds of vessels out in the ocean. They're always sending out these little automatic identification signal radio identifiers, which are then snooped on by these fleets of satellites. And then they do a whole lot of processing. They pick out which one of these are coming from fishing boats. And then they actually use some artificial intelligence algorithms in order to figure out when they're fishing and when they're not fishing. Anyway, it's a fascinating data product. But this is what the global fishing effort looked like over, I think this is 2015 to 2017, where the more orange colors here are showing more intense fishing effort from this global satellite product. And drawn on here are some gray lines. Those show the exclusive economic boundary outlines. And often within those, people don't use these AIS transmissions. There are some biases in the data set. But I'm also showing on here in blue the outlines of the HNLC regions again. And I think it's obvious if you look at it that in general, the HNLC regions have pretty low fishing effort compared to the others. And when we compare that, so if I just average the global fishing effort within HNLC regions versus non-HNLC regions, it comes out as 0.05. So 1/20th. So if you just don't know anything else about the ocean, you just compare HNLC regions versus non-HNLC regions, this estimate from Global Fishing Watch gives you 1/20th of the fishing intensity inside HNLC regions. So it seems to be a pretty remarkable difference. And we wanted to test that against the expectations of a simple model. So, well, it's not that simple, but a model where, a model called BOATS, where we predict fishing and fishing activity from net primary production and water temperature. So in each grid, so we split up the whole ocean into grid cells. In each 1 by 1 degree grid cell, we look at the water temperature and the primary production, and then it's optimized against observations in order to estimate the fishing effort that you would expect given those two environmental forcings. And so when we look at this globally tuned model, which is tuned incidentally in large marine ecosystems, so those are coastal ecosystems, when we look at what it predicts for the whole open ocean, we get the top right panel here. Oh, sorry, this is in Frontiers in Marine Science. It's not in review anymore. We get what's shown on the top panel here, where it looks kind of okayish, maybe, overall for large parts of the ocean, but clearly not getting the low fishing effort in the HNLC regions. And then when we put in a simple, like a very simple schematic iron limitation dependence, we get something which looks much better. Which is, so agreeing with the low fishing effort in the HNLC regions. Okay, wait, I thought I went a little fast through that part. Any questions about any of that so far? At that point?
I have a question.
Yes, go ahead.
Okay. If the iron to carbon ratio is low in the zooplankton and the fish need more iron, why can't they just eat more zooplankton?
I mean, it's a great question. So I don't really know. But why? I mean, why do people get anemic? People also get anemic. We live in a very iron-rich environment compared to HNLC regions. I mean, there's so little iron in HNLC waters that it's extremely difficult to measure, as Chris well knows. So I don't know. It may be that it's just the way that life functions. There's only a certain amount of flexibility and how much material you can process. Like it may just be that organisms are geared up for this whole kind of production chain of breaking things down and using them to reconstruct new things, and you can't just flood the system with more material. That would kind of be my guess. Or it could be an ecological thing that is just, if you can't grow fast enough, you just get outcompeted by other things. And so other organisms that don't have high iron requirements win, and epipelagic fish are the losers. I don't know. I think I think there are many unanswered questions here. Thanks for that one. Any other questions right now? Okay. So finally, so if you don't believe this, because I mean, I have to say, like, I was kind of skeptical of this all the way through as well, but then I found one thing that just made me a believer in this, which was the ice fish. Does anybody here know about the ice fish? So this is a group of natothenoids. These are teleosts in the Southern Ocean. So one group of natothenoids, I don't even know how to pronounce this, the Canichthiidae, but these are known as white-blooded ice fish. And they were described like a century ago as this mysterious, bizarre organism. This is the only known vertebrate of any kind that has ever given up hemoglobin. So I haven't really talked to this point about what iron is good for in fish, but one of the main things is that it's used in hemoglobin and in myoglobin as well, which is basically the same thing, but it just sits in your muscles as an oxygen transporter. So this is the only known vertebrate that has given up hemoglobin entirely. And it's difficult to give up hemoglobin, apparently, if you're a vertebrate. In order for this organism to function with its greatly reduced oxygen transport capability, It's had to quadruple its heart size. It has a much higher volume of blood than similar sized fish of any other kind. It's sluggish. It's terrible. It's a terrible organism. It barely functions. And yet it's dominant in the Southern Ocean. The Tothanoids are the dominant Southern Ocean teleos. There's like nothing else once you get off some little parts of the shelves. And it's been a mystery for 100 years why these things persist. I mean, this was so great. I had to take this extract out of this review paper from 2006, that the existence of these fish appears at odds with modern evolutionary theory, which suggests that selective pressure should lead to the retention of hemoglobin and myoglobin. And people have actually held this up as an example of bad evolution, of evolution gone wrong because there were no competitors or something. I mean, I don't really follow the line of reasoning, but so it was suggested that this was some kind of colossal evolutionary mistake, that they just lost their hemoglobin and couldn't get it back again. So I would suggest instead that this is actually an adaptation to the extremely low iron concentrations. The Southern Ocean is the world's largest iron-limited HNLC region, and I think it's entirely consistent with the scarcity of iron there, that this would encourage the evolution of these hemoglobinless, relatively low iron requirement fish as a result. So I think so this is all, I mean, it's definitely a hypothesis. I don't know how to prove it, but it definitely would benefit from further testing. But I would just put this as a hypothesis that the iron availability plays a significant role in the abundance and distribution of marine fish to a degree which remains difficult to perfectly assess. Right. I said I was only going to talk for half an hour, and that was already half an hour. But so I'll just say a couple quick words about the return impact that fish could potentially have on the iron cycle. And here, I'm not necessarily thinking about adult fish. And I don't know, maybe you could even kind of blur this and think about it as being important, as kind of including zooplankton in now as well. So really thinking down to quite small fish, but non-microscopic animals. So maybe kind of generalize this part to large animals, where large means non-microscopic. So if we go back to this cartoon again, so we've got here the ingested iron to carbon in the food. So a little bit of orange, quite a bit of blue, that goes through the organism. The organism absorbs a large fraction of the carbon. So it's usually absorbing something like 80–85% of the carbon in its food. It doesn't assimilate all of that because it burns most of it off to support its metabolism. But it's taking that much carbon out of the food, but it's just getting a little bit of the iron, because the iron is much more difficult to get across the gut wall because of the difficult chemistry of iron. And so then this would predict that you would have high iron to carbon ratios in your adjusted fecal matter on the right. So more orange in the adjusted part compared to the ingested part. And so I think that's very important, that animals should tend to absorb most of their carbon, which they then burn up for energy to support their metabolism, and they fail to absorb most of the iron. So they're stripping off carbon, but most of the iron is passing through the gut. So what should this do? This should enrich the ingested material. This is enrich the feces compared to the food source in a very consistent way. So is this observed out in the ocean? Well, there's not much data on it, but there is some data on it. People have measured everything. And usually in the 70s, actually, we found when we went out looking for these weird things to measure, people usually measured them in the 70s. And then we're like, that's interesting. And then they stopped. but so there is some data out there. And it's kind of all over the place. So this is showing the iron to carbon ratio of the feces compared to the iron to carbon ratio of the food. There are very few paired measurements. Those are the green ones. So that's where actually somebody measured the food and measured the feces and compared the two of them. More often it was unpaired. We had to try to guess at what the thing was. Oh, and sometimes there were stomach paired. So they would look at what was in the stomach, measure what was in the stomach compared to what came out in the feces. Anyway, this gave a range of ratios going from 1 up to something like 100, but nothing really that was significantly below 1. So consistent with there always being an enrichment, and typically enrichments of something like 5 to 10 probably. I don't know if you put something in the middle there, something like 5 to 10, but that's about what you would expect from the ratios I was talking about before, the assimilation ratio of like 0.1, 0.15 for iron compared to carbon, which is like 0.85. So something of that order of magnitude. So if you then add this up, if you say, okay, we know that fecal matter is a contributor to the sinking flux. So iron is always being removed from the surface of the ocean and transported down to depth by sinking particles. This is the main reason why iron is in such low availability in the ocean. It's always being stripped out in these sinking particles. We know that these feces contribute some amount to that sinking flux. We know that these feces should be enriched relative to their food by some amount. We can then say, okay, how much should the overall sinking flux of organic iron be enriched by this fecal carbon stripping process, which we call the fecal pump? Just kind of gross, but I don't know, we couldn't think of anything else. So this fecal pump could contribute an enrichment. Well, so if we start on this plot, and this is just a very simple calculation, this plot. This is saying if there's no fecal matter in the sinking flux of organic particles, then it's just 1, there's no enrichment. This is the enrichment of particles on the vertical axis here. And then that could go up to something like 20 or 40 at the very high end where it's dominated by feces. But the most likely line here is the black, and the most likely fecal matter contribution to export is probably something like 20 to 40%. So you could see it's probably, this would say it's probably something like a 3 to 6-fold enrichment in the sinking flux of iron compared to carbon in many regions. But so pretty significant. So likely a multiple fold enrichment in the iron richness of sinking organic particles in the ocean, which is not usually considered. It's most often considered, I mean, it's very difficult to measure this because it's hard to separate out the organic fraction of iron, as I'm sure Chris could tell you much more about than me. But it's very hard to separate that out and look at it exactly. And so it's hard to know how important this is overall, because there are also abiogenic iron sources within the sinking flux, but this should be significant to some degree and will probably vary regionally quite a bit. And then finally, one more important thing about the sinking flux is that this then serves as food. I mean, almost all of the feces are eaten again, and then more feces produced, and those are eaten too. I mean, really, most of this stuff cycles through a few organisms. So I won't explain this calculation, but it's in Priscilla's paper, which just came out a few months ago. And basically, but it's a straightforward idea. This is just that every time an organism is eating the feces of another organism, it's benefiting from that enrichment in the iron. So you'd expect that coprophages should be able to kind of soak up that iron that's leaking through the planktivores. And that actually, because the mesopelagic ecosystem is so dominated by coprophagy, that it may never be iron limited. So it may be that these iron limited regions I was making a big deal about for the epipelagic fish that we like to eat in HNLC regions, maybe underneath that you have mesopelagic fish that have no problem with iron. I think that's an interesting possibility, because it does seem like mesopelagic fish are quite abundant in those regions. Just a couple more speculations because it's kind of fun. If you think about the life cycle of fish, you would expect, well, so livers basically build up a store of iron. Iron is toxic, so you don't want too much of it, but we can store it in ferritins, I think they are, in the liver. And so you can build up a reservoir of of iron in your liver, which can be like a many month or even like a year-long supply. We cycle through it quite slowly. But juveniles should be really susceptible to iron limitation, even on short timescales, because they're rapidly growing and they don't have the reserve built up of iron. So it's possible that this could drive life history strategies, such as salmon. Salmon do very well in the subarctic North Pacific, strongly iron limited. Why is it that they go into streams and rivers to reproduce? I emailed some salmon specialists and they're like, we don't know, they just do it. So could it be that they go into these very iron rich environments in order to spawn, because their juveniles have particularly high iron requirements? And then once they've stocked up a liver supply of iron, they can head out into the open ocean and take advantage of that huge pool of iron poor zooplankton, which are sitting offshore. One possibility. And finally, if you think about endotherms as compared to ectotherms, we have a very different assimilation efficiency for carbon, because we burn so much carbon in order to run our high temperature machinery. We have very low assimilations for carbon compared to ectotherms. And so as a result, we actually automatically enrich our iron contents compared to our food. So rather than being a ratio of 1, as we saw for the fish, this is much more likely to be a ratio closer to 5 or something. And so we should actually do better on iron poor food than fish could do. And this may contribute to the high abundance of endotherms in HNLC regions as well. Who knows, but a possibility. Okay, so finally, iron limitation is well established for phytoplankton, but has not been considered significantly for animals beyond zooplankton. When you look at the elemental ratios and assimilation efficiencies, it suggests that many, if not all, fish should struggle to survive in iron poor HNLC regions. And indeed, fishing efforts are anomalously low in the HNLC regions. Then the fecal enrichment of iron can potentially make a major contribution to the global sinking flux of iron. I think we need better measurements to know for sure, but it sure seems likely. So overall, iron availability may play a major and previously unrecognized role in the ecology of large marine animals, and animals may conversely play a major role in the global iron cycle. Thanks very much. I'd be happy to take any further questions.
All right, thanks so much, Eric. Very interesting. So I have a question, but I'd like to open up to the audience for questions first, and pick your method of asking the question. You can type your name in the chat or raise a hand, or I think there's a few enough people you could just go ahead and unmute and break in. But do we have any questions from the audience? All right, I'll start with my question. So you showed that the fish tissue was in general higher in iron to carbon than copepods, even though they were supposed to be poor at assimilating the iron. So is it that they actually have some kind of iron concentration mechanism we don't understand, or is it that we don't fully understand what the fish are eating if they're eating these iron-rich fecal pellets, or getting that through a food web?
Wait, sorry, what did I show? That the
you had a plot with iron to carbon ratios, and I believe the fish tissue had higher iron to carbon ratios than copepods or zooplankton. Is that right?
Wait.
Or you were suggesting that they had higher iron requirements than the
was
food
it this
available.
plot? This plot?
It was, yeah, this one.
Okay.
So the salmon have higher iron to carbon than the zooplankton.
They're higher than the zooplankton in iron limited waters where measurements are available. So Priscilla did a huge amount of work on this table. Like, many of those circles are from individual studies where they just had one or two measurements. So it's putting together a bunch of studies, but we grouped them so that iron-rich waters are all in the top section there. and then iron limited waters are in the middle. So, but the zooplankton are shown in both, right? So the phytoplankton are in both, the green bars, and the zooplankton are in
Right,
both,
right.
the brown bars. So, and basically they overlap. So the zooplankton are just not very different than the phytoplankton. So iron-rich
Okay.
waters, you've got iron-rich phytoplankton and iron-rich zooplankton. So it's it should be no problem in those regions for fish, I would think, right? In coastal waters, there should be, maybe in, I guess like in California where you have iron limited waters upwelling, maybe it's not the same, but in many coastal and estuarine waters, I would assume there's plenty of iron in the zooplankton. You can get values up to 200. I mean, Hutchins showed values of like 150 or 200 micromole iron per mole carbon in coastal waters. So that should be fine. It's really just a very, very different out in the open ocean where the zooplankton are hugely flexible. They can really, I mean, they don't have the complicated needs for iron that fish do. So they can go down to these extremely low iron to carbon ratios, apparently.
Okay.
That makes sense? Yeah, I should have explained this figure a little bit better.
No, that makes sense.
Okay.
And I have a related question, if I may.
Sure.
Eric, that was great. I enjoyed your presentation a lot. That was really good.
Okay,
So I was thinking about the fish now, and from the evolutionary standpoint, it seems that the fish have not evolved to make the best out of the iron conditions they have, in that they don't seem to take advantage of limiting iron concentrations in the most efficient way they could, right? I mean, you think about number one, the alkaline gut. I mean, I didn't know that. And then the processes, I don't know the physiological processes, but it seems to me that the evidence points that the fish have not evolved in the most efficient way to utilize the iron around them.
Yes, so that's a great question. And I suspect that's probably part of the reason why there hasn't been, why people haven't proposed this before from like the ecology side of things. Like I came at this from a biogeochemistry side, because I worked on HNLC regions before. And then actually I started working on this because I was looking at the global fishing maps and I was like, what the heck's going on in the HNLC regions? Why is there no fishing? That's actually how this whole thing started. But so I feel like that, like people in the ecological world are so used to seeing great adaptations to things that there's almost this implicit assumption that everything has adapted well to the existing environment, which makes you feel like nothing should be limiting in a way. And yet, I think the way that it could have come about like this is that fish, I was trying to look into this actually. I think teleosts developed in fresh waters. I don't know if anybody knows about that here. I could not find a straight answer on that. It seems like it's perhaps even hard to say, because I did find some papers that suggested that teleost evolution had kind of gone back and forth between marine and freshwater environments. Mm-hmm. But it may just be that they did not start out as an open ocean organism. Instead, they started out in freshwater environments. And so they kind of developed all their physiology for an iron-rich environment, so that they weren't being cheap with iron. And then once they got to the ocean, I mean, it's very hard to get rid of hemoglobin, as the Antarctic ice fish shows. Like that's, you can't just like flick a switch and suddenly have, you know, a different, like change your electron transport chain, or like you have these built-in iron requirements, and it may just take massive evolutionary changes in order to reduce that, because it's such a profound change in the physiology that's required. Mm. And at the same time, there's other organisms out there. So it's not that there's nothing living in the Southern Ocean. Actually, I also found a paper about how the Southern Ocean has cephalopods that take the role of fish. That was like the title of the paper. It's like squids take the role of fish in the Southern Ocean. And cephalopods don't use iron because they don't, in the same way, because they don't have hemoglobin, they have hemocyanin, actually use copper as well. And that'd be an interesting thing to look into, maybe. Copper availability versus iron availability, I don't know. But so it could just be that it's not like there was this vast open niche that was not being utilized. It was just being utilized by different organisms. And that when the teleosts came to those iron limited regions, they were not able to displace the other things from the epipelagic environment. that make sense?
Yeah.
I'm a little out of my depth there, but that's how I kind of rationalize it to myself.
Well, I'm with you. I mean, the reason why they have not evolved is because something else is more constraining, right? Yeah. So, and you know, and fish move. I mean, fish move. You think about they can exploit micro areas with perhaps more iron than others. I mean, they can move, they can travel miles if need be. And we know that iron distribution is not homogeneous, right? So they may exploit these little pockets.
Absolutely.
I know, I'm thinking a lot here.
No, absolutely. And I showed this to a tuna expert, and he got very excited because it's unexplained why tuna tend to migrate to waters around Indonesia, the equatorial Pacific tuna. They often go to Indonesia in order to spawn, which was not understood, but the waters around Indonesia are actually very iron rich. So, who knows if it explains that, but I do suspect that this could be one aspect of what's driving some life history strategies of fish. And we shouldn't be surprised. Like, it's like, we know that humans need micronutrients, and there's there's a lot of concern about micronutrient limitation in poorly nourished human populations. Like, it's a real concern for us, including iron. Iron's one of the big ones.
That's great. One more good question, if I may. I'm sorry, I'm taking a lot of time. Sure. How much of a problem is the alkaline gut? Is that a big problem, or you think there is something else physiologically that is the bottleneck as to why these guys need so much iron?
Right, that's a great question. Actually, I almost took that slide out because I got really excited about that when I first discovered it. And then I talked to Rod Wilson, who was the author of that paper, and he said it's still unclear. Who knows how much of a problem that is. I mean, I tell you one thing, I really came to appreciate how difficult digestion is working on this project. We kind of don't think about it. We just shove some food in and then trust that it's going to get taken care of and we'll be fine. But when you get into the chemistry of digestion, holy smokes, there's a lot of stuff going on in there. There's all these microenvironments that are being delicately maintained. And it's really a very difficult chemical process that's going on in there. So I don't know for sure, but it just seems that iron is quite difficult for anything with a gut to assimilate. It's a tough, it seems to be tough to take. The organisms can definitely downregulate it, that is for sure. Organisms that have too much iron can downregulate their absorption of iron because it's toxic, and apparently few of it, or maybe no organisms have mechanisms to get rid of it. Once you've got the iron, you're kind of stuck with it, and it takes many months or years to flush through your system. So you don't want to have toxic levels of iron, but it seems like there's this upper limit to how much iron can be assimilated through a gut, which is quite low.
Thank you.
You're welcome.
All right, any other questions?
I have one more.
Sure.
What on the evolutionary timescales of fish, what has been the iron availability in the ocean? Were there times when there was more dust and more question. Iron input?
So I would love to see reconstructions of the last glacial maximum when there were when there was greater dustiness in the atmosphere, and when we think the HNLC regions probably shrank. I tried to get some people to dig through sediment cores to find fish debris, which you can do, but it's hard to get material in the right places. If you go further back in time, actually the Southern Ocean probably became its modern self, with the Antarctic circumpolar current about 30 million years ago. And I would assume that that's when it first became this big HNLC region. And that is, as accurately as can be determined, the branching time of the Notothenoids from other teleosts. So the beginning of that group really coincides with the opening of the Southern Ocean, which is today the world's largest HNLC region. It could be that the subarctic Pacific and the Eastern tropical Pacific are quite ephemeral. I mean, that's another reason why there maybe hasn't been a big evolutionary impetus for fish to become better at low iron concentrations, because they may not always have been so extreme. We may be living in a world with particularly large HNLC regions. I'm not sure. Something I've wondered about.
All right, we still have a couple minutes before 12. Anybody else has something else?
Doesn't sound like it.
Okay, well, yeah, if not, let's all thank Eric again. And, we can close the formal seminar so people are excused. If you don't mind staying on, Eric, I know there was at least one person who wanted to say hi, and anybody else is welcome to stay on a little longer to talk in a smaller group. But thanks again.
Sure.
Thank you.
Okay, thank you. Thank you.
Thank you.
Well done.
Okay, bye for now.
Bye, guys.
So Eric, I'm sorry for my question, but just to go over this again on this slide.
Oh, is it still up? Oh yeah, sorry.
Yeah, the salmon, Southern Ocean fish, those ratios are higher than the zooplankton in the iron limited waters. But your
Yes.
point was that they kind of overlap. Is that what
Yeah.
you were saying?
That's right.
Okay. I think I was thinking like they do look a bit higher. So how are they getting more iron than food?
Oh, I
And
see what
maybe
you mean.
maybe that we don't know exactly what they're eating.
Right. So, oh yeah, okay. Yeah, that is confusing, isn't it? So I kind of like,
But there's a flexibility there that we can not be so strict about exactly where they are.
I kind of stand way back and squint at this figure.
Okay.
Like those, so these are salmon from aquaculture. These are whole fish measurements of aquaculture
Okay.
salmon. So
Right.
who knows what they're like actually. I mean, I would assume they're kind of the same. I don't know for sure.
At least, yeah, in terms of orders of magnitude, it's, yeah, I see.
And they're Atlantic. These are Atlantic salmon in aquaculture. And the Southern Ocean fish were not open ocean. They were from, if I remember correctly, I think they were from coastal Antarctica and I think from a subantarctic island. So,
Okay.
whereas the zooplankton there, I think those are from like the full-on iron limited part, I think, if I remember correctly. But really, like, if there should be giant error bars on these, we just didn't even know how to put the error bars. So I would say they kind of like overlap-ish, the low end of the fish range.
Right. Okay.
With the iron limited range, but maybe not. I think that there really should be specific, like you really have to go out and like measure what the food is, measure what the fish are. It's hard.
Yeah, I was thinking about like if it's not a linear food chain, if it's a web, then yeah, it might be hard to map that out completely.
It really might be.
But yeah, let me introduce Diana Bernstein. She's a atmospheric scientist and climate modeler, and yeah, she wanted to say hi.
Oh, hi.
Hi.
And you sat through all the fish stuff.
Yeah, I'm actually aerosol modeler, so this is how I interested in iron.
Okay.
So I don't know if you know more about this, but there's our recent studies show that there is we understand a little bit more how iron comes to the ocean. That it's not only from the dust, but most of it in the remote ocean comes from the fire aerosols as a dissolved iron.
I've heard that, but you say it's most of it?
Not most of it, but the big most of it in the remote ocean.
In the remote ocean. Yeah, that's very interesting. That's very interesting.
And I actually was curious, and I guess Steve was asking this question about the paleo scale. And I was just curious, again, as a modeler, I'm curious how you can actually find the evidence in the paleo scale. You said you probably from the course you can learn about what happened, about the fish.
Yeah, so I mean, so sediments, ocean sediments are basically the garbage can of the ocean, right? So a little bit of what's going on up above always